Good afternoon and welcome to my educational presentation on the chloroplast, mitochondria, their origin, creation, and misconceptions that surround both of them. We do have several objectives today, one of them which is the creation of the mitochondria and the chloroplast. This has long been a subject of discussion among scientists. I will attempt to clarify the accepted way that these organelles were created. Next, we will move on to organelles and the needs of the plant. Fact that both are needed, the mitochondria and the chloroplast, in their cells to function properly. I will explain why this is possible and has to happen. There are several misconceptions about the mitochondria and the chloroplast. I will attempt to inform you why these are false ideas about the mitochondria and the chloroplast. And the general target audience for our lesson today is going to be an introductory level undergraduate student in general biology. What is the importance of what we're actually doing here? Knowing where these two organelles come from is a key to understanding how they work and why both are found in plant cells. The function of the plant requires that both the mitochondria and the chloroplast are needed to function, survive, and thrive. We're going to look to dispel several misconceptions in regards to these two organelles. I will attempt to dispel them about these two organelles in a clear and concise manner. As we begin our introduction to the unit, what is this about? This is about the idea that plant cells need both the chloroplast and the mitochondria, and how there are misconceptions regarding this issue. Why is it important? The importance is that each of these organelles do for the plant cell and what each one's function is to work together. Why should we explore this? Well, we need to explore this so that we can understand the function of cells and organelles in plants and their proper functioning. To begin with, we need to talk about the endosymbiotic theory and its origins. Dr. Lynn Margulis was the one that first came up with this idea. This was in the 1960s, and the picture on the right is actually a picture of Dr. Margulis. Her book, titled Symbiosis of the Cell, in 1981, became the official recognized ideas behind the endosymbiotic theory. We're going to continue by talking about the organelles in the beginnings. The beginning was a prokaryote merged to live together with a larger cell that we call a eukaryote. This merging led us into the action of the organelle, and this action comes from the creation of free living bacteria that were absorbed by early eukaryotes and mixed with other DNAs, created what we believe were the organelles we call the chloroplast and the mitochondria. The action of creation was really the merger of cells to make two specific organelles, the mitochondria and the chloroplast. Concluding on this construction phase, these two organelles have their own DNA genome, they make their own proteins, they continue by dividing existing mitochondria and chloroplast to continue survival. And finally, the last step in this aspect of the endosymbiotic theory is that the ingested prokaryotes developed into organelles we are discussing today. Once ingested, the eukaryotes, the organelles that formed, can no longer live outside of the eukaryotic cell. As we look at the origins of the endosymbiotic theory, there are prokaryotic hosts. We're going to model some chloroplasts and mitochondria formations, and we're going to look at step-by-step creation. First off, the origin of the endosymbiotic theory indicates a prokaryotic host that becomes part of the eukaryotic cell as organelles by endocytosis. Looking at the diagram on the right-hand side of the page, you can see the path that a prokaryotic cell takes to become part of the eukaryotic cell's organelle, specifically the mitochondria and the chloroplast. You can follow along the chart here and see how they have become one within the new eukaryotic cell. In this step-by-step depiction, you see the origin of the prokaryotic host, then the various stages of transition, then the proteobacterium into the eukaryotic cell, and finally the transfer of genes from the organelle to the nucleus of the eukaryotic cell. Now, we've just discussed the origins of the endosymbiotic theory. The origins of the mitochondria are a little bit different. To understand the creation of the mitochondria, you need to refer to the endosymbiotic theory that bacteria were partially responsible for the creation of the mitochondrial organelle in eukaryotes. Again, if you look off to the right here in the white section of the screen, you will see over time a more complete mitochondria has been formed as more and more bacteria become involved and then completely absorbed into the eukaryotic cell, thus completing the cycle that ends up with a mitochondria in the eukaryotic cell. Looking at the bottom, you see the gamma proteobacteria, 89%. Moving up, the beta protobacteria, 95.2%. Continuing moving up, you see the alpha proteobacteria at 93.8%. And then finally, you see all of those have merged over time, 100% mitochondria. The origins of the chloroplasts are a little bit more difficult to understand, but it follows the same general guideline. As with the mitochondria, the same process that leads to us is the endosymbiotic theory. However, the size of the genome length in a chloroplast in comparison to frequency compared to the mitochondria shows that the gene frequency in the chloroplast is higher than that of the mitochondria. Thus, this is critical to the new eukaryotic cell. The chloroplast can pass on the genetic material for the new cell. Taking a look over here on the right-hand side, you see under letter B, the chloroplast genome length is longer than the genome length of the mitochondrial. As we continue into now development of our chloroplast, we talk about the post-endosymbiotic merge, continued bacterial involvement, and the peptoglycan covering dealing with both of these. Again, as with the mitochondria, the endosymbiotic theory dictates the growth and development of the chloroplast. The development of the chloroplast includes the continued involvement of bacteria to aid in the development process. The peptidoglycan covering is a result of phagocytosis, or the engulfing of the double membrane system after the endosymbiotic reaction has occurred. The PGN may be ancestral and is now common in eukaryotic cells. If you take a look off to the right here in the diagram, you can see the PGN, also the peptoglycan, merging and creating an outer layer on the cellular structure. Following the cursor, you can see me circling the beginning, then you see the chlorophyll in the purple, and then you see the merge in the pink color. The beginning of the merge on letters B through E are one species of algae, and the beginning of the merge on F through I is a separate species of algae. As we continue once again, the development of the mitochondria is also a result of that endomembrane theory. Once again, we're going to talk about the endomembrane theory in regards to development of the mitochondria, but we're also going to talk about its evolution into other species. The evolution of the mitochondria took different steps to get in the diversity of plants and species. Looking at our diagram off to the right, you can see the humble beginnings of the mitochondria, then the evolution as it moves up the tree to chloroplastidia, right here on the left-hand side, following the blue line, then to land plants as well as other marine plants. Land plants is this green line here right by the cursor, and then as you see moving up and up into different species altogether. Along the same lines, you will see a progression into rhodophytes, and off to ciliates and other varied species. Ciliates here, rhodophytes over here. This evolution to other species is made possible by the mitochondria's ability to split itself, reproduce, and have a completely new mitochondria in the cell. The big part of these misconceptions is, why are both of these actually needed in plant cells? Energy capture and carbon fixation is done by the chloroplast, and then energy conversion, true energy for the plant, is done in the mitochondria. The chloroplast is needed to capture the incoming solar radiation. Then carbon is converted to an organic molecule so that it can be used by the plant. The mitochondria is needed then to convert the solar energy collected by the chloroplast into ATP, adenine triphosphate, and ADP, adenine diphosphate, for use throughout the plant. Without both of these organelles, the plant could not survive. With only the chloroplast, energy would be collected but not converted for use by the mitochondria. Thus, no energy for the plant. With only the mitochondria, there would be no way to convert the collected solar energy into ATP or ADP for plant usage. Looking at the top picture on the right-hand side here, you can see a chloroplast in its normal state, indicated by the labels of collecting light energy and then converting it for use by the mitochondria and then the plant. Looking at the bottom picture, you can see that the mitochondria is in a normal state as well, indicated by the labels showing it is converting the transferred energy from the chloroplast into ATPase for use within the plant. Having gone over the background of development, why they're needed, origins, let's talk about the misconceptions that are out there. Misconception number 1. The mitochondria is not needed in the plants. The idea behind this misconception is that the idea that a chloroplast converts energy and thus powers the plant with light energy. Secondly, the chloroplast has heteroplasmy, which is the presence of multiple copies within the cell. Thus, the abundance of chloroplasts has led to the misconception that it is the only needed power source for the cell. And as you can see off to the right here, this is a picture of a mitochondria just loose within the cell. Misconception number 2 is that energy in plants is not from the mitochondria, and plants can grow without oxygen. The misconception that energy for plants does not come from the mitochondria. The mitochondria, however, is the key player in the energy cycle for plant cells, including the processing of an energy in a hypoxic environment. This means an environment with low oxygen levels. An example of hypoxic growth due to the work of the mitochondria are tightly packed seeds. They can use limited oxygen and increase levels of nitric acid to grow. The picture off to the right shows the actions of the mitochondria using little oxygen to convert energy and thus create the nitric acid. You can see here, as things are going through the mitochondria, out comes the nitric acid up here at the top, and going in you see the hydrogen ions, and then coming out you see NADPH, glycolysis taking place, the fats have broken down, and again the mitochondria is actually helping a plant grow in a limited oxygen environment. Misconception number 3. The mitochondria is just extra. That's not really the case. The mitochondria has a central position regarding metabolism. They help provide energy as well as a metabolic go-between to the cell and other regions. The mitochondria, with its own proteome, a set of proteins that can be expressed by the genome, organelle, or other tissues at certain times play a key role in apoptosis or scheduled cell death as needed by the cell. Without the mitochondria, there would be issues with the elimination of cells from the plant body, and there could be an abundance of dead tissues in the cell, causing additional problems. The actualities of these two organelles, the truth, both are needed in plants. This was stated by Lynn Margulis, and we have learned through research that they are symbiotic and use symbiosis to grow and develop. Both the mitochondria and the chloroplast are needed for the plant to survive. The combination of these two organelles provides the proper energy so the plant can function and operate properly. Lynn Margulis, approximately 50 years ago, proved that the chloroplast and the mitochondria were formed from the simple phagocytotic ingestion of prokaryotic cells, making them part of a larger eukaryotic cell. For many years, her work was rejected without even having been read. Now her work is the gold standard for the endosymbiotic theory. Symbiosis in the cell is vital to its survival. The mitochondria and the chloroplast share that symbiotic advantage. The symbiosis of these two organelles has existed since the beginning of eukaryotic cell advancement, and it appears that it will continue, for both are required to provide energy to the plant cell. To tie everything together, a quick discussion in regards to the endosymbiotic theory, chloroplasts, mitochondria, and misconceptions. As you have seen throughout this presentation, there is a lot of material that has been covered. Don't worry. We will build on this and review this throughout the entirety of plant units that we are covering. There are origins that were in question. Thanks to Dr. Lynn Margulis, those origins have now become the standard for the development of chloroplasts and mitochondria in plant cells. The development of the mitochondria and the chloroplasts came from prokaryotic cells that were engulfed into eukaryotic cells and became organelles, and we see this by the double membrane as one of the key ideas. The combination of the chloroplast and the mitochondria are required for plant cells to collect light energy in the chloroplast and then transfer that energy to the mitochondria, where the light energy is converted to use usable energy for the plant to survive. As you can see here in the picture, we have the prokaryotic cell moving over to the modern host. You see these loose cells of prokaryotic genomes, data, whatever you'd like to call them, but you see them move into the modern host cell, and you see they have now formed the organelles, the chloroplast and the mitochondria. The misconceptions that the mitochondria are not needed in plant cells was debunked. Evidence was presented that energy for a plant is from a combination of the chloroplast and the mitochondria, and that the mitochondria is not just an extra organelle, it is part of a larger system to provide energy to the plant. Finally, there are facts to support the statement that the mitochondria and the chloroplast are both needed to allow the plant to function and survive. Are there any questions on our topic here today? If not, we will be finishing up our presentation here. Just a quick review. This slide here is a list of the references that you can access for additional information on each of these. Here's the next set of references, again, with their direct links so that you can learn more, especially in this case about Dr. Lynn Margulis and the groundbreaking work that she did. And finally, our last few references, which shows everything from modern day to worldwide acceptance of the fact that chloroplasts, mitochondria are both required for plant cells to survive. Thank you for watching this presentation. I hope you enjoyed it, and we will move forward with this as we continue on the unit.